Biomaterials, especially polymers, are used extensively in drug delivery technologies. Hence, it becomes imperative to study their compatibility in living systems. A plethora of materials such as lipids and polymers are used to encapsulate drugs of synthetic or biological origin to produce therapeutic nanoparticles like the COVID-19 vaccine, and then their pharmacology and toxicology is assessed. However, the US FDA provides certain guidances to study the safety of excipients in preclinical animal models before their clinical application. In our case, we will study the effects of a biopolymer trimethylchitosan in animal model. Hence, the objective of my study is to develop innovative methods to study the long-term fate of biomaterials in vivo. Our laboratory specializes in radiolabeling different materials, producing nanoparticles, injecting them in vivo, and then assessing biological samples to study their effects for short duration of time. But the question arises, what happens when these materials are injected, allowed to circulate for a long period of time, and then evaluated? The possibilities are that the nanoparticle could remain unchanged, or it would exist as non-degraded excipient, or form degradation products and liberate the free label. To assess all these conditions, we made use of chromatography that separates the high molecular non-degraded products from the low molecular degradation product. To establish the proof of concept in vitro, we radiolabeled TMC with tritium, subjected it to different degradation conditions, and assessed the chromatograms. We found that 95% of the polymer underwent degradation when in contact with a concentrated acid after 72 hours. In case of an enzyme, 33% of the biopolymer was degraded, and when in contact with macrophages in cell lines vitro, we found that 46% of the polymer degraded within 72 hours. Next, we injected rats with radiolabeled TMC nanoparticles and found that these were rapidly cleared from in vivo and distributed in the liver and kidneys. Upon assessment of the chromatographic analysis, we found the presence of large molecular weight and small molecular weight species in both these organs at the end of 24 hours. In another experiment, we placed rats in metabolic cages and collected excrement over 14 days. We found that 15% of the radioactivity was eliminated in the feces and 10% eliminated in the urine. Upon chromatographic analysis, we found the presence of both large molecular undegraded and small molecular degraded products over the period of 14 days. In a nutshell, we were able to combine the techniques of radiolabeling and chromatography to assess the biodegradation of a material in vivo and found that TMC could be eliminated and biodegraded in rats after 14 days of injection. Thank you for your kind patience.